In the last video I covered an overview of making proteins from DNA, and then I covered transcription in some detail. In this video I will cover translation, and then I will also talk about some mutations that occur to DNA and connect those mutations to the processes of transcription and translation. So when I ended the last video, we had produced our mRNA from our DNA. How do we make a protein then from that mRNA? Well, first we need more RNAs. So we start with our mRNA, we need to add our transfer RNA, or tRNA, and ribosomal RNA, or rRNA. So if our messenger RNA, our mRNA, has the code to make the protein, our tRNA, or transfer RNA, is the RNA that carries the amino acids that are going to be used to make our new protein. Now, each tRNA carries a different amino acid. The tRNAs all have the same basic structure, but there's some variability in the loops. Here's three different ways you'll see tRNAs depicted. Here is a two-dimensional schematic of a tRNA. Here is a more representative three-dimensional depiction of tRNA, and then here is a very simplified schematic that you'll often see used, particularly in this textbook. So the loop at the bottom of our tRNA is called the anticodon. The anticodon is complementary to the codons that are found on the mRNA. And remember, those codons on the mRNA are three bases that each code for an amino acid. There are 61 different codons that code for amino acids. Remember that three codons are stop codons. And so out of our 64 possible combinations of bases, 3 of them are stop codons. There's 61 that code for amino acids, but only 45 different tRNAs exist. That's because the third position of our anticodon is said to have wobble. So tRNAs, these anticodons, the first two positions bind very faithfully to the mRNA, but that third position doesn't bind with quite as much precision. And so there's 61 different possible codon sequences that code for amino acids, but we only have 45 tRNAs that recognize those codons. The ribosomal RNA is found in the ribosome, as the name suggests. Both ribosomal RNA and tRNA are made from DNA. Remember, we started this unit talking about the idea that DNA makes RNA, RNA then makes protein. But that's only kind of accurate. Not all RNA then becomes protein. Some RNA stays in the cell as RNA and is used as rRNA or tRNA. I gave an example in the last video of how some RNA is used as an enzyme in the spliceosomes. So our rRNA is again found in the ribosome. The ribosome has two subunits. It has a large subunit and a small subunit, and that ribosome has 3 slots for tRNAs. It has an A slot, a P slot, and an E slot. That E site is the exit site. That's where our tRNAs are going to leave the ribosome after they have been used. The P site is going to hold our growing polypeptide, and the A site is where we bind what we call our aminoacyl tRNAs. These are tRNAs that are still attached to an amino acid. So now let's look at how this process of translation occurs in the ribosome. Translation begins when the small ribosomal subunit is going to bind to the mRNA, and the mRNA is going to bind to the initiator tRNA, the tRNA that binds to the start codon. Remember, our start codon is a sequence AUG, going from the 5' end to the 3' end of the mRNA. So the anticodon on the initiator tRNA goes from the 3' end to the 5' end with the sequence UAC. So this initiator tRNA binds to the start codon. That complex binds to the small ribosomal subunit. And then the small subunit plus the mRNA plus the tRNA all together are going to cause the large ribosomal subunit to bind as well. And so now we've completely assembled our ribosome. I'm going to walk through now the elongation of that growing polypeptide using a figure from the textbook, but the figure has already assumed several steps have occurred and aren't depicted in this figure. So what I'm going to call step 0 is the slide I just talked about, where when the ribosome forms, we have the tRNA that has methionine the initiator tRNA located in the P site. So this initiator tRNA has recognized the start codon. That initiator tRNA has methionine bound to it, and it's going to be in the P site of our ribosome. We then have a new tRNA that's going to come into the A site. The tRNA that comes into the A site is going to be dictated by the next codon on our mRNA, so these 3 bases right here. This is an endergonic process. It requires an energy investment. That energy comes from the hydrolysis of GTP. We then are going to have a new peptide bond that's going to transfer the growing polypeptide chain that is located on the amino acid in the located on the tRNA in the P site. That amino acid will be transferred to the tRNA that is in the A site. This reaction is going to be catalyzed by the ribosomal RNA. The addition of another GTP, so we have GTP hydrolysis again, we have another endergonic reaction that's going to cause a shift of tRNA from the A site to the P site. So now the tRNA in the P site has that growing amino acid chain, and then the tRNA that was in the P site gets shifted to the E site and is going to exit the ribosome, leaving now an open spot in the A site for the next tRNA with the appropriate amino acid to come in and continue this cycle. This process will continue until the ribosome encounters a stop codon. The stop codon binds a release factor. This release factor is a tRNA-like protein. So it is a protein that has a shape similar to a tRNA, and that shape allows it to bind in the A site of our ribosome. So that release factor is going to come in, it's going to recognize one of the stop codons, and that release factor is going to promote the hydrolysis of the bond between the tRNA in the P site and the last amino acid in that growing polypeptide. And so we're going to break that bond, we're going to release our tRNA, we're going to release our free polypeptide, and then the ribosome is going to break apart and can be used now to start this process all over again with a new piece of mRNA. One last point I want to make about this process before we move on is that mRNA is often translated by many ribosomes at once. So what you're seeing here in this scanning electron micrograph are a whole bunch of ribosomes that are all translating one mRNA at the same time. So you can see this mRNA strand running through the middle and all these ribosomes that are translating that mRNA all at the same time. Now, if you'll recall, when we talked about cell structures, I talked about the idea that the endoplasmic reticulum comes in 2 different types. We have a smooth endoplasmic reticulum and a rough endoplasmic reticulum. The difference between the two is that the rough ER, the rough endoplasmic reticulum, has ribosomes attached while the smooth ER does not. Right? Ribosomes can either be free in the cytoplasm or they can be attached to the endoplasmic reticulum, and the endoplasmic reticulum with those ribosomes is our rough endoplasmic reticulum. Right, I'm going to end this video talking about mutations in our DNA, how those mutations are going to affect amino acid sequence, and we're going to connect that back to this process of transcription and translation. So in the last unit, I talked about large-scale mutations that occur on chromosomes. But way back in the first unit, I gave an example of how a small-scale mutation can affect protein function. I talked about sickle cell anemia. And this sickle cell anemia disease is due to a single nucleotide change in our DNA that leads to a single amino acid change. That single amino acid change leads to a large change in protein structure and function. Right, so we have this one amino acid glutamate is inserted into the normal protein because of the nucleotide sequence in the DNA. Changing that nucleotide sequence causes a valine to be inserted instead of that glutamate or glutamic acid. Right? It causes a difference in the charge of that hemoglobin molecule. Right? It goes from being a negatively charged amino acid to a nonpolar amino acid. That's going to cause a difference in the structure of that final functional protein, which is going to cause a difference in how those proteins sort themselves in the cell or distribute themselves within the cell, and that's going to cause a difference in cell shape, which causes a difference in how well red blood cells bind to oxygen. So let's look at some of these mutations that occur to DNA. Let's start with this DNA sequence. We have the DNA sequence where we have the 3' end, then we have TAC, TAA, GAG, TTT, and here's the 5' end. So I want you to pause this video and make a protein from this section of DNA. Right? You need to be able to identify whether this is the template or the non-template strand of DNA. You need to create the appropriate mRNA, and then you need to use the one-letter amino acid abbreviations, such as this F for phenylalanine or this S for serine, and the appropriate codons on your mRNA to make a amino acid sequence that will be part of our growing polypeptide. If you made the amino acid sequence from the nucleotide sequence on the previous slide, you started with this nucleotide sequence, and hopefully you were able to identify this as the template sequence. It was the 3' end of our DNA going to our 5' end. That means you would make the non-template sequence of DNA, which would be complementary to the template sequence, and our mRNA then would look like our non-template sequence, except everywhere there is a T in our DNA, there is a U in our RNA. And so we can then take this mRNA and we can make our amino acid sequence. So our peptide is going to be formed first with these first 3 bases, AUG, and that codes for methionine. The next codon, AUU, codes for isoleucine. The next 3 bases code for leucine, and then the last 3 bases, the last codon, codes for lysine. So we have the 4 amino acids that are represented by the letters MILK. You just made the protein coded by this nucleotide sequence here, this DNA sequence. Now I want you to make the amino acid sequence that is coded by this DNA sequence. If you transcribed and translated that protein, hopefully you created this non-template DNA that's complementary to the template sequence. You then should have created this mRNA where we have U's in the mRNA, everywhere there is a T in the non-template DNA, and then the peptide sequence, you would start with the same codon, the start codon AUG that codes for methionine. Even though we've had a mutation in our template sequence, you still code for the same amino acid isoleucine with the next codon. Even though we had another mutation in our DNA, we have the same amino acid coded by the codon. And even though we had a mutation in this last codon, right, we again produce the same amino acid. So even though we had these changes in our nucleotide sequence, we didn't change our peptide sequence. These changes can go by several different names. So we did a substitution. We replaced one base pair with another. So for example, we replaced what used to be an adenine with a guanine. So we had a substitution. That's also called a point mutation because it was a change of one nucleotide. So we had 3 different point mutations. Right? We had a point mutation here where that adenine was changed to a guanine. We had another point mutation where a guanine was changed to an adenine. We had a third point mutation where thymine was changed to a cytosine. Each of those mutations was also a silent mutation. They were silent because they had no effect on that final amino acid sequence. All right, here again we're back at the original sequence that you translated that spelled the sequence of amino acids MILK. Now I want you to transcribe and translate this DNA sequence. So if you transcribed and translated that nucleotide sequence, you first got this non-template DNA sequence, you then got this mRNA sequence, and then you use that mRNA sequence to create the peptide with the amino acid M, because the first codon still represents methionine. The next codon is going to code for the addition of isoleucine. The next codon is for leucine, but that last codon now is going to code for a different amino acid. It used to code for lysine. We used to have a TTT, which coded for AAA on the non-template strand, or AAA on our mRNA, which then produced a lysine amino acid, or resulted in a lysine amino acid being added to our growing polypeptide, and now we've changed that to a glutamic acid. So we still have a point mutation, right? We changed our T to a C, we changed one nucleotide, but this is now a missense mutation. It's a missense mutation because we've changed the amino acid sequence. Here's another mutation for you to work through. So here was the original sequence that had the amino acids MILK, and now we're going to make this an amino acid sequence from this nucleotide sequence. So if you transcribed and translated this DNA sequence, you got this non-template DNA sequence and this mRNA sequence, and then when you made your peptide, you started with methionine, and then isoleucine, and then leucine. But now that last codon is going to be a stop codon, so it's going to stop translation. When you have a mutation that leads to the insertion of a stop codon, we say you have a nonsense mutation. A stop codon has been added to the sequence. You're going to create a non-functional protein because you're going to stop adding amino acids to that polypeptide sequence. This is again a point mutation because we've only had a change in one nucleotide. It was originally a T, now it's become an A, so we've had a point mutation, and this point mutation is also a nonsense mutation. Here's another sequence of nucleotides for you to transcribe and translate. Again, here's our original, and now transcribe and translate this sequence where we have had a nucleotide added to the sequence. So here's the TAA in the original, followed by a GAG. Here's that TAA, here's that GAG. We've just inserted an adenine in the middle of that DNA sequence. If you transcribed and translated that nucleotide sequence, you should have gotten this non-template sequence and this mRNA sequence, and that leads to a peptide that begins with methionine, then is followed by isoleucine. Now we have a missense mutation. What used to be an L is now an S, so it used to be a leucine is now serine. And then we have a glutamine inserted after the S. So we've had what's called a frame shift mutation. We added a base pair. A frame shift can be an addition or a deletion of a base pair, and that shifts the reading frame downstream of that mutation. All right, we still have the same sequence of nucleotides after the mutation, but now we've shifted the reading frames because instead of this GAG being transcribed and converted into the codon CUC, two of those nucleotides are part of a different codon. So we've had a shift in the reading frame. The codons now are being read differently. That same sequence of nucleotides is being read differently within our mRNA and during translation to create a completely different sequence of amino acids. All right, let's do one last one. So here what I've done is I've taken the original sequence and I've inserted 3 nucleotides into the middle of that sequence. So here's your TAA, here's the GAG, and then I've inserted the CAG in between those two sequences. So if you transcribed and translated this last nucleotide sequence, you should have gotten this non-template sequence and this mRNA sequence, and then when you made your peptide, you started with methionine, then isoleucine, then valine, then leucine, and then lysine. And so what we've done here is we've added 3 nucleotides, and that 3 nucleotides results in the insertion of a single amino acid. Everything downstream of that is unchanged. Right? We started in the original nucleotide sequence, which was translated to a peptide that was MILK. We still have that LK after our mutation. We've just inserted one whole amino acid. You could also delete 3 nucleotides if we had cut out, without inserting these nucleotides, if we had cut out these 3 nucleotides, right, you would have just skipped that leucine and would have continued on the same reading frame. So a 3 base pair change is going to lead to the insertion or deletion of an entire amino acid. So in this unit, what I've talked about is how we go from our DNA to our polypeptide. But every cell is different. Some cells express some parts of DNA, other cells express other portions of the DNA, different genes. So in the next video, what I'll talk about is how does the cell determine which parts of the DNA are going to be expressed and which parts are not going to be expressed.